Hi there. Today I'd like to discuss a topic of natural products that is, you can say, farnesol. There is an example of sesquiterpenes. Sesquiterpenes means the terpenes which contain three isoprene units in its structure. Those are known as, you can say, sesquiterpenes. An example of sesquiterpenes, you can say, farnesol. Farnesol, from this term, from this word, we are guessing the, you can say, a functional group: farnesol, alcohol, methanol. That means all. This term all, in case of chemistry or organic chemistry, all, we know that about all, this term was added or address alcoholic group. Farnesyl all that addresses a alcoholic group: methanol, ethanol. That means we are guessing the functional group that is attached in this, in its structure. It could be that, you can say, alcoholic group. It could be, you can say, primary alcohol, secondary alcohol, or tertiary alcohol. It doesn't matter. We will probe the overall structure. We will elucidate the structure of farnesyl, and along with this, we will know about the stereochemistry if it's possible for today. Farnesyl, that is a, you can say, it is a acyclic sesquiterpenes. This acyclic sesquiterpenes occurs in the well of ambrat seeds. We will found this farnesyl, ambrat seed, rose, and citronella, lime flowers, severely orange. Actually, this kind of, in case of these plants, will be able to extract farnesyl terpenes, and as a form of oil or as a form of essential oil. And that is the second portion. We will elucidate the structure of farnesyl, or we will be able to constitute the structure of farnesyl in this specific portion. Scientist Kirschbaum in 1913 elucidated or established the structure of farnesyl. The molecular formula, C15H26O. That is a, you can say, molecular formula. You can ask me how they are able to elucidate the structure or molecular formula of farnesyl. We know that about, for any kind of chemical compound, we'll be able to find out that, you can say, empirical formula for different from different grades of analytical data. We'll be able to find out the empirical formula for we able to know about the find out molecular formula from any kind of empirical formula. So in this way, they have found the molecular formula of farnesol was C15 H26O. And this, we can say, structure elucidation or structure establishment by Kirschbaum in 1913. And the second part, that is called, you can say, presence of three double bond. Actually, Kirschbaum in 1913, he have said that, actually, in case of, in the structure of farnesyl, there are presence of three double bonds. How will be able to find out the presence of three double bonds in case of farnesyl structure? According to the knowledge of chemistry, according to the knowledge of organic chemistry, when uh, we'd like to do a hydrogenation, or you can say, when I'd like to do bromination or halogenation, we'll be able to find out the existence of double bond or existence of unsaturation. If we add 1 mole of hydrogen in case of any compound, then we'll be able to see that when 1 mole of hydrogen, if it was added, then we can able to say that actually the compound contains only one double bond. When 3 mole of hydrogen will be added in a single compound, that then we can say that that single compound will contain three double bond. And when we can say that, you can say, similarly, we can say that when 4 mole or 5 mole hydrogens, if it added, then we can say that four and five double bonds is present in that specific compound, or in this in that specific structure. So in this way, we'll be able to see that presence of three double bond we can prove in the following way. C15H26O, when you'd like to add three hydrogen, then we can say that C15H26. That means hydrogen is 26. When you would add it 3 hydrogen, then we can say that it's a 26 plus 6 hydrogen will be, you can say, 32. And oxygen will be as usual, as starting material or starting compound. And as 6 hydrogen, 3 mole of hydrogens means, you can say, 6 hydrogens are added in the farnesyl. So we can say that hexahydro, because 6 hydrogens are added in this portion, we can say that it's a hexahydro farnesyl will be produced, and the produced compound name will be hexahydro farnesyl, because 6 hydrogen atom attached to this farnesyl molecule. Similarly, we can prove the existence of 3 double bond by means of bromination. When we will add 3 mole of bromine, that means 6 bromine uh, atom, this farnesyl group, then if it added properly, then we can say that the existence of 3 double bonds is present, or you can say 3 double bond is present in the, you can say, farnesyl compound or farnesyl molecule. We have added 3 mole of hydrogen, 3 mole of bromine, that means 6 bromine atom. Then we can add it C15H26O Br6, that means 6 bromine atoms are added. We can say similarly the name of the produced compound will be hexabromopharnesol, because 6 bromine atom attached to this farnesyl. That, so we can say that the produced or synthesis compound name will be hexabromopharnesol. And the third portion, at first we have find out the molecular formula. Secondly, we have actually confirmed the presence of 3 double bond. And thirdly, we will confirm the, you can say, functional group of farnesyl. Presence of 1-degree alcoholic group. Actually, the scientists have established the structure or elucidated the structure of farnesyl by means of following this, you can say, steps. Presence of 1-degree alcoholic group. We know that about this is an, you can say, you can say, symbol of 1-degree alcoholic group. Uh, Alcoholic group. So we can say that it's a primary alcohol or 1-degree alcoholic group. And we know that about alcoholic group or primary alcoholic group. When you'd like to oxidize a primary alcoholic group, it will be converted into aldehyde group or CHO group. So he have written, we can written, we have written C15H26O, C15. We can write it down, C15. It is a C15H26O. We can write it down, this molecular formula, C15H26O, C14, and this carbon, it will be the C15H23H2, that means H25, and H1, that means you can say that total number of hydrogen is H26, and remaining oxygen will be as usual. That's how we can say that C15H26O, that is the molecular formula of farnesol. We can write it down in the following way. We would like to show this oxygen as a, you can say, egg degree or primary alcoholic group form, and the remaining portion will be attached as a, you can say, alkyl chain. When we have, when you'd like to probe the existence of 1-degree alcohol or primary alcohol, when we'd like to you can say, oxidize this primary alcoholic group containing compound or farnesyl, then if it contains a primary alcoholic group, obviously it will be converted into aldehyde group. And from this conversion, we are seeing that CH2OH that has been converted into CHO containing compound, that is called, we can say that it's a farnesyl. It could be the, you can say, farnesyl. We know that about, in case of aldehydic homologous class, the, you can say, in the prefix portion L, methanol, ethanol, propanol, L is added. That's why farnesyl, it will be converted into farnesyl, L. This L is added due to the presence of this aldehyde group. Actually, as farnesyl is converted into farnesyl, we can say that in our compound, in our farnesyl compound, obviously, actually 1-degree alcoholic group is present. Actually, otherwise it couldn't be converted into farnesyl. And the next one, that is called, you can say, some degradation products of farnesyl, possible structure of farnesyl. How we can degrade, we can convert, it's a farnesyl when you'd like to oxidize in the presence of chromic acid or chromium trioxide, then we can say that it will be converted into farnesyl. That means this 1 degree alcoholic group CH2H will be converted into CHU. Actually here oxidation is occurred when you'd like to convert, when you'd like to do a reaction in the presence of, it is called hydroxylamine. OH means hydroxyl, NH2 means amine. We can say that it's a hydroxylamine. Then it will be converted in, it will added with this compound, and it will be converted into CH double bond O. Actually, aldehyde group is present as a CH double bond O. This C double bond O, this oxygen will attach to this hydrogen, and it will be converted into water, and it will be converted into water, and remaining portion will be, we can say that C uh, double bond. Actually oxygen is eliminated, that's why we can say that carbon contains free two double bond, or you can say double bond. This double bond will attach with nitrogen, or remaining portion of hydroxylamine, and it will be converted into CH double bond and OH. That is called the, you can say, farnesyl oxime. uh, when you'd like to, you can say, uh, pass in the presence of dehydrating agent. Dehydrating agent means the agent or chemical compound that causes dehydration. That means here is a hydroxyl group is present, here is another hydrogen group is present. When you'd like to add this hydrogen with this hydroxyl group, it will combine with each other and form odor. And that's why HOH together form outer, and C double bond N will be generated, C double bond N, C triple bond N, because when H will be eliminated from this carbon, when OH will be eliminated from this nitrogen, then one single bond will be free, one single bond will be free on the nitrogen, and this free single bond will be connected with each other and form a triple bond. That's why we can say that C14H23 C triple bond N will be created. That is called, you can say, farnesyl nitrile. That is called, you can say, farnesyl nitrile, or uh, you can say farnesyl nitrile, or there is a, you can say, cyano compound or nitrile compound. So by means of dehydration of farnesyl oxime will be converted into farnesyl nitrile. When you'd like to do in the presence of potassium hydroxide and H plus, when you'd like to do alkaline hydrolysis, then you'll be able to convert these carbon triple bond N will be converted into CWH, or C14H23 will be as usual. That is called, you can say, farnesic acid, and the remaining portion uh, will be converted into C13H22 O. That means when you'd like to keep, when you'd like to assume two farnesyl nitrile, then obviously one mole will be converted into farnesic acid, and another mole will be converted into, you can say, C13 C13H22 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 O. That is a, you can say, ketone or ketonic structure, and the remaining portion will be converted into, you can say, carboxylic acid, or you can say, for from farnesyl nitrile will be converted into farnesic acid, because farnesic, it's a farnesyl nitrile. When it will converted into C triple bond will be converted into CWH. That means we can say that it's a oic acid, or you can say it's a carboxylic acid. That's why the name will be converted into as the fashion of carboxylic acid nomenclature, farnesic acid. And the next portion, that is called, you can say, ketone structure 15C13H22O. Ketone was found similarly to dihydropseudoinon. Dihydro pseudoinon, that is called the, you can say, geranyl acetone. That is called the geranyl acetone, uh, and confirmed by its preparation. Actually, how you will be able to convert this compound, or how you'll be able to synthesize this structure 5 compound. It's a geranyl or geranyl chloride. When you'd like to add with this, you can say ethyl acetoacetate, sodium ethyl acetoacetate, then it will be added with each other. And this Cl, you can say it's a geranyl part. This portion will attach with this, you can say, uh, carbon hydrogen, and this carbon hydrogen will attached with, you can say, this carbon hydrogen will attached with this C double bond, COOET, and this COOET will attached with COCH3. This will connected with COCH3. As COCH3, there is a acetone that is that would be connected with this CH bond, this CH portion. And in this way, geranyl chloride will connect it with sodium acetoacetate. And when you'd like to do hydrolysis, decarboxylation will occur. Here is a, you can say, COO ethyl, ethyl acetate is attached. So this COO will be converted into, it will be eliminated, and that's why we can say that it's a decarboxylation will occur, and the remaining portion will be as usual. During the formation of structure 5, there is a structure 5. You can say it's a C13H22O. That is a, you can say, ketonic compound. During the formation of 5, two carbon atoms are lost from the precursor. Characteristic, that is the characteristic reaction for alpha beta unsaturated carbonyl compound. Because here is a, you can say, COO. It's a, you can say, carbon. Here is another carbon. Two carbon has been lost, actually. That's so we can say that uh, it is a characteristic reaction for alpha beta unsaturated carbonyl compound. It is a alpha beta unsaturated carbonyl compound, alpha beta. When you'd like to do alkaline hydrolysis, then it will be converted into RCR double bond O, RCO, CH will be converted into CH3. Thus, to explain the degradative products 4 and 5, farnesol was considered as one. It's a farnesol. That is a, you can say, considered as a one. When you'd like to do oxidation in the presence of chromic acid, it will be converted into farnesyl. CH2OH will be converted into CHU. When you'd like to do in the presence of hydroxylamine and acetic anhydride, due to the presence of hydroxylamine, it will be converted into, uh, you can say, farnesyl oxime. When you'd like to, you can say, when you'd like to pour acetic anhydride as a dehydrating agent, here dehydration will occur and it will be converted into nitrile compound. When you'd like to do, that is called the, you can say, alkaline hydrolysis, it will be converted into COOH, and it is called the, you can say, geranyl acetone. This portion is called the, you can say, it is this portion is called the geranyl. This portion is called the, you can say, acetone. That is called the, you can say, geranyl acetone. Evidence in favor of structure I. Structure I means, or structure on farnesyl. In case of evidence in favor of structure on, we can say that it's a farnesyl. When you'd like to do ozonolysis, it will be converted into, you can say, glyceraldehyde. You can say glyco glycoaldehyde or glyceraldehyde. We can say that. We know that about the compound containing carbon-carbon double bond. Then obviously it will show the ozonolysis reaction uh, in the methyl sulfide, or in the presence of gene. Then this, when you'd like to break down this double bond, then obviously here will be attached with, you can say, ketone, or you can say here obviously it will be take a form, ketonic form, and here will be, you can say, it's a aldehydic form. Aldehydic form, it will be ketonic form. And that's why we can, when it like to break down here from, it's a, you can say, ketonic form. See, it's a, you can say, this is the, you can say, it's a hand, it's a, you can say, one carbon, it's a, you can say, ketonic form. Here will be the, you can say, 1 2 3 1 2 3. This will be the, you can say, CHU. CHU. Why it would be CHU? Because we know that about, in case of ozonolysis reaction, when carbon-carbon double bond containing carbon does not attach with any kind of alkyl chain, that it will be converted into CHO. And when carbon-carbon double bond containing carbon attached with a, you can say, alkyl chain or aryl chain or any kind of chain, then it will be converted into ketonic form. And similarly, we'll be able to see that it's a, you can say, ketone. Here will be, you can say, ketone as usual. It will be converted into, you can say, it will be converted into CHO, and it will be converted into, again, it's a CH3 CO CH3 acetone. That means we can say that it's a glyceraldehyde will be produced from this portion. It will be glyceraldehyde. From this portion it will be glyceraldehyde. From this portion it will be labelaldehyde. From this portion it will be labelaldehyde. That means two molecules of labelaldehyde will be converted. We can say that in the following, it's a number 1 carbon, it's a number 2 carbon, uh, it's a number 3 carbon, it's a number 4 carbon, it's a number 5 carbon, it's a number 5 carbon. That's so we can say that it's a 2 oxo pentanal 1. That is called the, you can say, label aldehyde. It is a aldehyde compound containing, you can say, ketonic compound, ketonic group. So we can say that in when you'd like to read ketonic group in the prefix portion, you can say it's a oxo, 2 oxo pentanal on. That is called the label aldehyde. By means of ozonolysis reaction of farnesol, we are able to form two molecules of labelaldehyde and one glyceraldehyde and one acetone. In the last portion, final conformation, we will be able to confirm the structure of, you can say, glyceraldehyde, sorry, farnesol, by means of synthesis. That is called the, you can say, neurolidal. That is the neurolidal. When in the presence of acidic environment, when it will be converted into, you can say, it will follow the allylic rearrangement. By means of allylic rearrangement, it will be this double bond will come to this double bond, and this wedge group will come to this here, and it will be converted into farnesol. From neurolidal, by means of allylic rearrangement, it will be converted into farnesol. If you'd like to give the, you can say, isomers of farnesol, four important isomers of farnesols have so far been is isolated by various workers. You can say trans, cis cis, cis trans cis. It's a trans. How can you explain this is a trans? You have to cis trans isomers of, you can say, uh, identification. You have to check out the CH2 position. When CH2 position in, you can say, in the across position, then we can say that it's the trans. When CH2 position will be the in same side, then we can say that it is, you can say, it is a, you can say, um, it is a, you can say, cis form, cis conformation. When CH2 in the similar side, then we can say that it is a cis. When CH2 in the trans, then we can say that it's it would be the C, you can say, opposite position. Then we can say that it's a uh, actually this is a double bond. This is a CH2 group, this is a CH2 group, these CH2 groups in opposite portion. That's so you can say that it's a trans. It is a double bond. This CH2 is in the same side. That's so you can see that it's a cis, it's a trans. In case of this conformation, if you check it out, if you'd like to identify the cis trans conformation, here, this is a double bond. We'd like to check it out from here. 1 2 3 4 5. 1 2 3 4 5. There is a 2 isoprene in it. We don't like to check it out this portion. We'd like to check it out this 2 portion. It's a double bond. This is a, you can say, this is a methyl, this is a methyl, this is another methyl. So these two methyl in opposite portion. That's so we can say that first double bond is in trans conformation. This is a double bond. You can say this is a this is another methyl, that is in, you can say, opposite position. That's why we can say that second double bond is in, you can say, trans conformation. Trans conformation is can be written as E or E. and the cis conformation that can be written as Z. So we can say that it is the, you can say, fast, it is the it is the fast, it is the second. So these fast double bond, uh, actually CH2, this is a CH2 group that is attached with this carbon. This is a CH2 group that is attached with this carbon. These two CH2 group is in the same side, in the upper side of double bond. That's why it is a cis conformation. If you'd like to check it out, this double bond, it is a, you can say, uh, it is a, you can say, CH2 group. Here is a, another CH2 group is present. These two CH2 groups in the, you can say, lower side. So we can say that it, as two CH2 groups in, is it in the same side, or as two CH2 groups is located or situated in the same site, we can say that it is a, obviously is in cis conformation, cis conformation, or you can say that Z conformation. And the next one, trans cis. In the it is the, you can say, fast double bond. Um, it is a, you can say, it is a, you can say, you can say it's a, this CH2 is a cis conformation. This CH2, this, if you'd like to check it out, this is a, this CH2 in upper, this CH2 in lower, that is a trans. That means first double bond that is in trans conformation. Actually first and second, how we'll be able to check it out? Actually there is a functional group, that's why functional group, actually this double bond is closer to this functional group, that's why it is a, you can say, first double bond, and it is a second double bond. For it is a second double bond. In this way we'll be able to find out it is you can say what would be the first or what would be the second double bond. It is the first double bond, and it is in, you can say, a trans conformation. That's why we have written down is a trans means we can write it down, it's a E. Trans means we can write it down, it's a E. And the next one that is called, you can say, this double bond, it's a CH2. Here is a, you can say, another CH2. This is a CH2, this is another CH2. This two CH2 is located in the upper portion of this double bond. That's why we can say that it's a cis position. Cis, we can write it down is a Z. That's why it's a EZ isomers is present in this, you can say, conformation. Actually it is the, you can say, fast double bond, because this double bond is closer to this functional group. That's why it's a fast, it's a second, because it is far away from this specific functional group. So we can say that it's a cis, it's a trans. Where? Because it is a CH2 group, it is a CH2 group. Two CH2 group is located or situated in the upper side of this double bond. That's why we can say that it's a cis. And the next double bond, or in the second double bond, we are saying that it's a CH2 group, it's a CH2 group, it's the another CH2 group that is in, you can say, opposite direction. Actually this CH2 is located in the upper side of double bond, this CH2 is located in the lower side of double bond. So we can say that it is in trans conformation. So it is in trans conformation. Cis means we can say that the 2CH2 is located in the same side. When 2CH2 is located in the different sides, so we, in such cases, we can say that it's a trans conformation. In the fast double bond, we are seeing that 2CH2 is located in the upper side of double bond, so we can say it's a cis. In case of this portion, we are saying that 2 CH2, it's a XCH2, it's a another CH2, 2 CH2, 2 CH2 are located in the opposite direction. That's why we can see that uh, second double bond is in trans conformation. We can write it down in the following way. Cis means we can say it's a Z. Trans means we can say it's a E. It's a Z-E isomer. So we can say that there are four isomers in case of farnesol. Trans means EE, cis or Z-Z. Trans cis, or we can say EZ, cis trans or ZE. Trans means E. Trans means E. So some special reactions, if you'd like to give some reactions of farnesol, potassium, potassium that is called the, you can say, potassium hydrosulfate, that, that is work as a, you can say, dehydrating agent. Dehydrating agent means this potassium hydrogen sulfate that will be able to eliminate odor, so odor molecule. as it will be able to eliminate the outer molecule, we can say that it is considered as a dehydrating agent, because it can remove the outer from the farnesol. In the presence of potassium hydrogen sulfate, when you would like to proceed a reaction, this hydroxyl molecule and from this carbon atom and another hydrogen will attach to each other, and it will be converted into water, and here another double bond will be generated, and another double bond is created, and from this specific double bond it will shift this here. That means here will be allylic rearrangement is occur. In this way, alpha farnesene is created. This is the, you can say, as usual, actually changes occur this double bond and this double bond. So guys, in the presence of potassium hydrogen sulfate, we'll be able to dehydration to farnesol, and we'll be able to produce alpha farnesene from farnesol. And the next one, that is called the, you can say, neroldiol, nerol nerol nerolidol. That is a, you can say, it's a double bond. It's a, you can say OH, nerolidol. When you'd like to do nerolidol by means of allylic rearrangement in the presence of acid, it will be converted into farnesol. When you'd like to uh, pass this nerolidol in the presence of potassium hydrogen sulfate, it will be converted into beta farnesene. It is called the alpha farnesene. It is called the alpha farnesene. It is called the, you can say, beta farnesene. Beta farnesene. So when you would like to do a reaction or dehydration reaction with nerolidol in the presence of potassium hydrogen sulfate, we will be able to found beta farnesene. When you'd like to do you can say dehydration reaction with farnesol, then we will be able to found alpha farnesene. In this way, we'll be able to uh, confirm the structure of, you can say, that is called the, you can say, farnesol, and we'll be able to identify different kinds of isomer of farnesol. So guys, this is all about today. Thank you for watching this video. Stay tuned, stay connected with this channel, hit the subscribe button and press the bell icon for watching such kind of videos in future.